Here we can look for the morphology of a bacteria. Bacteria, this is unicellular free-living microscopic microorganism which are capable of performing all the essential functions of life. They possess both deoxyribonucleic acid and then ribonucleic acid. Bacteria, they are the prokaryotic microorganisms that do not contain chlorophyll. They occur in water, soil, air, food and all natural environment. They can survive extremes of temperature, pH, oxygen and atmospheric pressure. Here we can look for the size of bacteria. Bacteria are very small microorganisms which are visible under the microscope. They are having the size range in microns. Bacteria are stained by staining reagents and then visualized under the high power of the magnification of the compound microscope. An electron microscope is used for the clear visualization of internal structure of the bacteria. Here we can look for the shape of bacteria. On the basis of shape, bacteria are classified as Kokai, Bacilli, Vibrios, Spirella, Spirochetes, Actinomycetes and the Microplasma. Next we can go for Kokai. Kokai, they are small spherical or oval cells. In Greek, we can say Kokou, which means berry. This is a micrococcus one. Bacilli, they are dot-shaped bacteria. Example is Bacillus anthracis. It is derived from the Greek word bacillus, meaning streak. In some of the bacilli, the length of the cell may be equal to width. Such the bacillary forms are known as cocobacilli, example Bracilla. Vibrios, they are comma-shaped curved rods, example Vibrio comma. Spirella, they are longer ridged rods with the several curves or coils. They have a helical shape and the rigid body, example Spirillum rubeum. Spirochetes, they are slender and the flexuous spiral forms. Actinomyces, the characteristic shape is due to the presence of rigid cell wall, example Streptomyces. They are branching filamentous bacteria, example Streptomyces species. While speaking about the Mycoplasma, they are cell wall deficient bacteria and hence they do not possess the stable morphology. They occur as round or oval bodies with interlacing filaments. Next we can go for the arrangement of the bacterial cells. Kokai appear as a several characteristic arrangement of a grouping Diplococcus, Streptococcus, Tetracoccus, Staphylococcus and the Saccharinae. The first one is Diplococcus. They splint in one plane and remains in pair, example Diplococcus pneumoniae. Streptococcus, these cells divide in one plane and remain attached to form chains, example Streptococcus lactis. Tetracocci, they divide in two planes and live in groups of 4, example Gafica tetragena. Staphylococci, coccus cells divide in 3 planes in an irregular pattern. These cells produce the bunches of cocci as in grapes, example Staphylococcus aureus, Staphylococcus albus. Sarcinae cells divide in 3 planes in a regular pattern. These cells produces a cuboidal arrangement of group of 8 cells, example Micrococcus tetragena. Arrangement of grouping formed by the Bacillus species, Diplococcus, Streptobacilli and the trichomes. Bacterial structure, one is the flagella, villi, capsule, plasma membrane, cytoplasm, cell wall, ribosome, mesosome, inclusion and spores. Flagella, they are long, slender, thin hair-like cytoplasmic appendages which are responsible for the motility of bacteria. These are the organs of locomotion. They are 0.01 to 0.02 mm in diameter, 3 to 20 mm in length. The flagella are made up of a protein flagellin. The flagellum has 3 basic parts. One is filament, another one is hook and another one is the basal body. Filament is of the thin, cylindrical, long outermost region with the constant diameter. The filament is attached to the slightly wider hook. The basal body is composed of the small central rod inserted into the series of rings. The gram-negative bacteria contain 4 rings as L ring, P ring, S ring, M ring, whereas the gram-positive bacteria have only S and M rings in the basal body. Flagella, they may be seen on the bacterial body in the following manner. Monotrichous, these bacteria have the single polar flagellum, example Vibrio cholerae. Lophotrichous, these bacteria have the 2 or more flagella only at one end of the cell, example Pseudomonas fluorescens. Amphitraches, these bacteria have the single polar flagella or the tufted flagella at both poles. Across parallel serpents. Pediatriches, several flagella present all over the surface of bacteria. Example, Escherichia coli, Salmonella typhi. Pili of the fimbriae. Pili of the hair-like microfibrae 0.5 to 2 mm in length and 5 to 7 nanometer in the diameter. They are thinner, shorter and more numerous than the flagella. They are present only on the gram-negative cell. They are composed of protein known as pillin. They are unrelated to motility and are found on the motile and the non-motile cells. Fimbriae and pili. These two terms are used interchangeably, but they can be distinguished. The fibrae can be evenly distributed over the entire surface of the cell, or they occur at the poles of the bacterial cell. Each bacteria possesses 100 to 200 fimbriae. Pili are usually longer than the fimbriae and number only 1 to 2 per cell. So the function of the pili is they play an important role in the attachment to surfaces. Hence pili is also called the organ of adhesion. Plasma membrane. The cytoplasmic plasma membrane is a thin 5 to 10 nanometer. It separates the cell wall and cytoplasm. It composed of phospholipids, which is 20 to 30%, and protein 60 to 70%. The prokaryotic plasma membranes, they are less rigid than the eukaryotic membrane due to lack of sterols. Function: it acts as a semi-permeable membrane controlling the inflow and the outflow of the metabolites to and from the protoplasm. It provides the mechanical strength to the bacterial cell. It helps in the DNA replication. It contains the enzyme permease, which plays an important role in the passage of the selected nutrient through the membranes. Cytoplasm. The bacterial cytoplasm is the suspension of the organic inorganic solute in the viscous water and the solution. The cytoplasm of the bacteria differs from that of the higher eukaryotic microorganism, which is not containing the endoplasmic reticulum, Golgi apparatus, mitochondria and the lysosome. It contains the ribosome, protein and other water-soluble components and reserve material. In most bacterial extrachromosomal DNA, the plasmid DNA is also present. Mesosomes. They are the respiratory site of the bacteria. The mesosomes are attached to the bacteria chromosome and is involved in the DNA segregation during the cell division. They are predominant in the gram-positive bacteria. Nuclear. The bacteria chromosome. These are not surrounded by the nuclear membrane, so it is called nucleon. The bacteria chromosome are made up of double-stranded circular DNA. Intracytoplasmic induction. Many species of the bacteria, they produce cytoplasmic inclusion bodies which appear as a round granules. They are made up of either glycogen or starch. They are either glycogen or starch. They appear reddish when stained with the polychrome methylene blue or the toluidine blue. Bacteria synthesize the loose amorphous organic exopolymer which is deposited outside and tightly to cell wall called the capsule. Capsules may be composed of the complex polypeptide or polysaccharide. Water, which is 98%, is the main component of the bacterial. Bacteria capsule. Sometimes the capsular material is loosely associated with the bacterium. It can be easily washed away. The loose layer is called slime layer. Capsulated bacteria produce the smooth colonies, and non-capsulated bacteria produce the rough colonies on the surface of the agar media. They protect the cell from drying. They protect the bacterial cell against antibacterial agents and phages. Cell wall. The cell wall is a rigid structure. The cell wall is a rigid structure which gives the definite shape to the cell. Situated between the capsule and the cytoplasmic membrane. It is about 10 to 20 nanometer in thickness and constitutes 20 to 30% of a dry weight of cell. The cell wall cannot be seen by the direct light microscope and does not easily stain by different staining reagent. The cell wall of the bacteria contains the diaminophthalmic acid, muramic acid, and the decanoic acid. These substances are joined together to give rise to complex polymeric structure known as the peptidoglycan or the murine or the mucopeptide. Peptidoglycan is a major constituent of the cell wall of the gram-positive bacteria, whereas a gram-negative bacterial cell wall, its presence is only 5 to 10%. This is a chemical structure of the bacterial cell wall. N-acetyl-muramic acid is present, N-acetyl-glucosamine is present, peptide chain and the pentapeptide chain. A comparison of the cell wall of the gram-positive and the gram-negative bacteria. 20 to 25 nanometer, 10 to 15 nanometer. The cell wall is involved in the growth and the cell division of the bacteria. It gives shapes to the cell. It gives a protection to the internal structure and acts as a supporting layer. To prevent the rupture of the bacteria caused by the osmotic pressure, differences between intracellular and the extracellular environment. The cell wall is helpful to provide the support for flagella, to regulate the certain degree of a passage of molecules into and out of the cell. It serves as a site of attachment for most bacterial viruses. Ribosomes, they are the center for the protein synthesis. They are slightly smaller than the eukaryotic ribosome. The sedimentation constant is 70%. 70S ribosomes. These 70S ribosomes are made up of two subunits, namely large subunit 50S and small subunit 30S. During the active protein synthesis, the ribosomes are associated with the mRNA, and such associations are called polysome. Spores. The process of the endospore formation is known as sporulation, and it may take 4 to 8 hours in the vegetative cell. The endospores are thick-walled, highly refractive bodies that are produced one per cell. Each bacterial spore on the germination forms a single vegetative cell. Therefore, the sporulation in the bacteria is a method of preservation and not reproduction. Spores are extremely resistant to desiccation, staining, disinfecting the chemicals, and radiation and heat. They remain viable for the centuries and help the bacteria to survive for the long period under the unfavorable environment. The endospore can remain dormant for the thousands of years. The spores of all medically imported bacteria are destroyed by moist heat sterilization, autoclave at 121 degrees Celsius for 20 minutes. The vegetative sporulation. Bacterial sporulation, first we can go for the vegetative bacterium, which is a new one, and DNA go for replication. The septum forms here and the DNA translocates into 4 spore. Spore is engulfed, spore coat forms, mother cell